Assalamu alaikum dear students. I am Muhammad Rauf, lecturer Department of Zoology Cust. Welcome to the course Principles in Animal Life 2. This is lecture number 28. The topic is Translation in Eukaryotes. Discussing agenda for the topic is Introduction, Molecular components of translation. Lecture outcome. After watching and listening this lecture, students will know how the information stored in messenger RNA is used for the synthesis of a polypeptide chain. Introduction. Translation is the RNA-directed synthesis of a polypeptide chain. The genetic information flows from messenger RNA to protein through the process of translation. So translation is a process in which the information encoded in messenger RNA is used for the synthesis of a polypeptide chain, and this information in messenger RNA was originally copied to messenger RNA from the DNA through the process of transcription. Translation is the synthesis of a polypeptide using information in the messenger RNA. Now here you will recall the genetic code which is the triplet of bases that represent amino acids. So RNA consists of the triplets of bases. Those triplets of bases are identified by the transfer RNAs, and when the complementary bonding is associated between the triplet of base on messenger RNA and the triplet of base on the transfer RNA, then the amino acid is released, and the same process is repeated with subsequent triplets on the messenger RNA. Ribosomes are the factories. These are the protein workbenches. These are the sites where the process of translation takes place, and ribosomes are universal organelles. They are not bounded by any membrane, and they are present in almost all organisms, both prokaryotes and eukaryotes. As a molecule of messenger RNA is moved through a ribosome, codons are translated into amino acids one by one. So this is a messenger RNA, this is a ribosome, and there are 3 slots in that ribosome which we will discuss in the subsequent slides. This is the E site or the exit site where the transfer RNA exits. This is the peptidal site or the P site where a peptide bond is established between 2 amino acids, and this is the A site where the anticodon carrying transfer RNA comes first. The translators or interpreters are transfer RNAs molecules, each type with a specific anticodon at one end and a corresponding amino acid at the other end. So this is a transfer RNA. It has a clover shaped structure. On the one side is attached a triplet codon which is called anticodon. This establishes a complementary association with the codons which are present on messenger RNA, and once the complementary association is established, I hope all of you know about complementary association, and once it is established, then this ribosome, this amino acid is released. So these are a kind of translators. They read this message, understands it, and they interpret it and translate it in the language of proteins. Now, what are the molecular components of translation? A cell translates an mRNA message into protein with the help of a transfer RNA or tRNA. So tRNA plays a very important role here because the tRNA must interact on its two ends. On its one end, it must interact with the triplet codon present on the messenger RNA, and on the other end it must attach the correct amino acid. So it is bifunctional. Transfer RNA transfers amino acids to the growing polypeptide in a ribosome. Translation is a complex process in terms of its biochemistry and mechanics. It is one of the most energy consuming process that occurs in the cell. What next? Now, the structure and function of transfer RNA. Each transfer RNA can translate a particular messenger RNA codon into a given amino acid. Now, because there are a total of 20 amino acids, so researchers are of the opinion that there are 61 amino acid encoding codons. So there are 61 transfer RNAs. Others are of the opinion that transfer RNA can also interact with synonymous codons. The same transfer RNA have, but the interaction at the anticodon loop is necessary. So there are variable figures regarding the number of transfer RNAs. The transfer RNA contains an amino acid at one end, at that end, 3' end, it has an amino acid attached to it, and at the other end it has an anticodon loop which interacts with those codons in the messenger RNA. If that interaction, complementary bonding is established, then this amino acid is released. So here they have shown three different transfer RNAs and their structures. This is a two-dimensional structure, this is a three-dimensional 3D structure, and this is the one they have used in this book, Campbell biology book. So transfer RNA itself is a single RNA strand that is about 80 nucleotides long. So transfer RNA is 80 nucleotides long and it is a single strand. But here you can see a double-stranded transfer RNA. If you remember, when we were studying differences in the DNA and RNA, we said that RNA can be double-stranded. If RNA folds on itself and complementary bases faces each other, then they can establish hydrogen bond and the RNA can become double-stranded, but it is never helical. So transfer RNA is made up of 80 nucleotides. In 3 dimensions, transfer RNA is roughly L-shaped, where one end of the L contains the anticodon that base pairs with an mRNA. So in 3-dimensional structure you can see this is an L-shaped and its 3' end there is an amino acid attached, and on the other end there is an anticodon loop which interacts with messenger RNA. Accurate translation requires 2 steps. First, a correct match between transfer RNA and an amino acid. So first the transfer RNA brings amino acids to the site of translation. So first the transfer RNA must bind with a correct amino acid. Why I am saying correct? Because the transfer RNA contains an anticodon loop on its other end, which must be complementary to a specific codon in the RNA, messenger RNA, and the codons on the messenger RNA signifies amino acids. So this transfer RNA having a specific codon should attach a specific amino acid which represents the codon, and the attachment of amino acid with transfer RNA is done by an enzyme called amino-acyl tRNA synthetase. This is the enzyme which is responsible for the attachment. You can see here they have shown the enzyme amino-acyl tRNA synthetase, which is responsible for the attachment of amino acid with transfer RNA, and this reaction is catalyzed by an ATP, and once amino acid is attached with a transfer RNA, then we call this transfer RNA is charged tRNA, and this reaction is known as charging. The attachment of amino acid with tRNA is a reaction known as charging. Now first the transfer RNA must bind a correct amino acid, and second, the transfer RNA must interact correctly with the codons in the messenger RNA. Now, the structure and function of ribosomes. So here there are three different illustrations of the ribosomes. This is a computer model. You can see ribosomes are made up of this ribosomal RNA and an equal amount of proteins, and ribosomes have two subunits, a large subunit and a small subunit. So ribosomes are the protein workbenches. They facilitate specific coupling of tRNA anticodons with mRNA codons during protein synthesis. Why I am saying they are protein workbenches? Because in the factories there are benches where the workers work and they do their work similarly. Amino acid also, the ribosome also provides a site, a bench where the messenger RNA and the transfer RNA can interact with each other. The large and small ribosomes are made up of proteins and ribosomal RNA. A ribosome consists of a large subunit and a small subunit, each made up of proteins and one or more ribosomal RNAs. Ribosomal RNAs are also of different types depending upon their sedimentation. So it may be 16S or 5.8S, so they are of different types. In eukaryotes, the subunits are made in the nucleolus. The RNA subunits are synthesized in the nucleolus. Nucleolus is located in the nucleus. Ribosomal genes are transcribed and the RNA is processed and assembled with proteins important from the cytoplasm. So once the ribosomal RNA is synthesized from DNA, remember that the DNA does not encode just proteins, it also encodes other types of RNAs, transfer RNA, ribosomal RNA, siRNA, snRNA, microRNAs. All those RNAs are synthesized from DNA, but only messenger RNA is translated. The other types of RNAs are not translated. So when ribosomal RNA is transcribed from DNA, then they find their way to the nucleolus, and the nucleolus already has the proteins arrived from cytoplasm, and their aggregation takes place in nucleolus. Complete ribosomal subunits are then exported via nuclear pores to the cytoplasm. This is a computer construct of ribosome. This one is the large subunit, this one is the small subunit. The three sites E, P, A are located in the large subunit, and you can see here this is a messenger RNA and here is a growing polypeptide chain. In the second part of this lecture, we will discuss how this process takes place. So this is the exit site where the transfer RNA leaves the ribosome. This is the peptidal site. If the transfer RNA comes here, then the peptide bond between the amino acid carried by this transfer RNA is formed with the chain which is already developed. And this is the A site where the transfer RNA arrives first. Now remember that when the process of translation starts, the very first transfer RNA will enter P site, the very first transfer RNA, and after that all the transfer RNAs will enter A site. So remember, only the very first transfer RNA enters P site. And why this happens? Because when the translation starts, all those three sites are vacant, they are empty. So the first tRNA enters here, second tRNA enters here, then this ribosome shifts, it changes positions. So what will happen? The first tRNA which entered here will come here, the second tRNA which entered will come here, and this A site will be free again. So the remaining tRNAs will always enter A site, and there will be a displacement equivalent to a single codon or 3 bases, which provides a vacant slot for the incoming tRNA. So P site, peptidal tRNA binding site, holds the tRNA carrying the growing polypeptide chain. This it holds the growing polypeptide chain, and is it already discussed, holds the tRNA carrying the next amino acid to be attached to the chain. So here the after the first, the second tRNA, and till the end all the tRNAs will enter this side, and this is exit site where the tRNA is released. So a tRNA fits into a binding site when its anticodon base pairs with an mRNA codon. Here, if a complementary association is established, then this tRNA will be in a position to release its amino acid. The P site holds the tRNA attached to the growing polypeptide chain. You can see the tRNA located at P site is holding the growing polypeptide chain. The A site holds the tRNA carrying the next amino acid to be added to the polypeptide chain. This is the A site, and it is holding an amino acid, and it has established complementary association with the codons on the mRNA, and when these two slots are filled, then there will be a displacement. This ribosome will travel in this direction to 3 bases, and those 3 new bases will come in that position, and these two tRNAs will be shifted to E and A position, and then this one will start holding the growing polypeptide chain, and this slot will be emptied and available for the incoming tRNA. And this is the location where the discharged, means the tRNAs whose ribosome is, whose amino acid is removed, will leave from that position. Now the polypeptide grows at its carboxyl end, means there will always be a carboxyl end available which will bind with the amino end of the incoming amino acid. It's the end of part first of this topic. Kindly refer to the second video for the remaining part of the lecture. Thank you. Allah Hafiz.